What if we could read a single protein molecule from start to finish and detect disease before symptoms appear? At Northeastern, Professor Meni Wanunu is pushing the boundaries of biomolecular sensing through nanopore technology.
I'm Meni Wanunu. I'm a professor in physics, also in bioengineering. So my lab has two areas of focus. One area of focus is looking at the sequence of biomolecules using various methods. The main method currently used in the lab is nanopores. And nanopores are tiny holes through which biomolecules pass. As they pass, they produce electrical signatures, and we try to decode the sequence of molecules from those signatures. The second area of focus is using waveguides to look at enzymatic processes, and we're using an enzymatic process called reverse transcription in order to sequence RNA molecules. We are all made up of cells. Our cells have different types of biomolecules in them, and when some of those biomolecules are misbehaving or they have a different structure due to some erroneous processes that happen biologically, which are likely to happen, sometimes that causes disease. Detecting those molecules and being able to kind of classify the types of molecules that are present in a cell helps you to diagnose disease. The first step to doing that is being able to take molecules and reading them one by one and kind of saying where the errors are and where the aberrations are in the molecules. In order to do that, for DNA and RNA molecules, technology has been developed over the last decade. For proteins, this has not been the case, and methods are still being developed. And our lab is one of several labs around the world that are using nanopores to try to address this and try to solve the problem of reading a full-length protein molecule from beginning to the end and decoding its sequence and its level of modifications in order to see what type of disease might be present in a particular individual. To commercialize, a lot of money to develop the devices. So just to give you an idea, the type of investments that are necessary to bring to fruition biotechnological devices such as this one are in the order of hundreds of millions of dollars. Obviously, in an academic setting, even if I had that money today, I wouldn't have the infrastructure and resources to carry that forward. Where our lab is at right now, we are doing several areas of exploration in terms of developing the sensor itself, in terms of developing the platform that would be used to host the sensor, which is the nanopore itself. And then we're also looking for case studies, looking for areas where we think we can make a difference based on existing knowledge of proteins. So we are carving out those niches and being able to show that this works on a small scale would help us, carry forward into larger scale exploration. Yes, CRI has been very helpful. I've known some people at CRI for just about 13, 14 years since I joined here. They have helped me a lot with filing inventions, understanding, the market and where these inventions could be licensed to, and facilitating interactions with potential partners, potential companies that would license these inventions, and having discussions with me about commercialization, and basically from beginning to the end, they've been kind of, holding my hand in an area that I had very little experience with.